Picture this. You know exactly how a complex magic trick works on paper. The math is flawless, but you are completely unable to find the physical trapdoor on the actual stage. Well, that was exactly the frustrating, yet absolutely fascinating position that biologists found themselves in at the end of the 19th century. Welcome to this explainer. Today, we're going to unravel a true historical detective story. We are diving deep into the chromosomal theory of inheritance. This is the groundbreaking scientific journey that finally connected the abstract math of genetics to the literal, physical reality of our cells.
So our central mystery really kicks off with Gregor Mendel. Now we know Mendel essentially birthed modern genetics through his meticulous work with pea plants. He proved unequivocally that traits are inherited in predictable mathematical patterns. But here is the massive catch that completely baffled scientists for decades. What physically carries these traits? You see, Mendel was doing brilliant math, but biology is a physical science. What were these invisible blueprints actually made of? And where exactly were they hiding inside the organism?
Here is our roadmap for today's explainer. 1. Mendel's missing physical link. 2. The chromosome connection. 3. Meiosis, the genetic dance. 4. Comparing genes and chromosomes. And 5. Morgan's fruit fly proof.
Section 1. Mendel's missing physical link.
When Mendel published his famous laws of inheritance back in 1865, he proposed that distinct factors controlled traits, things like whether a pea plant would be tall or short, or have yellow or green seeds. He figured out that these factors occurred in pairs, that they segregated when reproductive cells formed, and that they assorted independently. The math was brilliant, but the term factor was essentially just a placeholder. He had absolutely no physical proof that they existed. I mean, he couldn't exactly put a factor under a microscope, right? Because of this, and a few other reasons, his incredibly important work sat largely ignored for about 35 years. Biologists needed to see the actual physical structures responsible for these traits before they were going to buy into the math.
Section 2. The chromosome connection.
Okay, let's dive into this timeline, because the way everything syncs up here is just beautiful. Fast forward to the year 1900. 3 scientists, De Vries, Correns, and von Tschermak, independently reproduce and rediscover Mendel's results. Suddenly, Mendel's abstract laws are the hottest topic in all of biology. But this time around, scientists have a major new weapon: vastly improved microscopes. By 1902, researchers are finally able to carefully observe the actual physical division of cells. And 2 scientists in particular, Walter Sutton and Theodore Boveri, notice something inside the cell that is behaving remarkably like Mendel's invisible factors.
These newly discovered microscopic structures were called chromosomes. That name literally translates to colored bodies, because at the time, scientists had to use these special chemical stains just to visualize them under the microscope. And as they watched these cells prepare to divide, they saw these colored bodies within the nucleus actually double, pair up, and divide. Sutton and Boveri realized these structured physical bodies were the prime suspects. They had finally found something in the cell that could theoretically carry Mendel's inheritance data.
Section 3. Meiosis, the genetic dance.
Now, let's break down the exact biological mechanism Sutton and Boveri were studying. You can think of the cell division process, known as meiosis, as this highly choreographed genetic dance. Imagine a cell with exactly 4 chromosomes, so 2 pairs. Step 1, the homologous chromosomes pair up right at the center of the cell. Step 2, during a phase called anaphase, these chromosome pairs are pulled apart. They segregate independently to opposite ends of the cell. Finally, step 3, the cell divides into germ cells or gametes. This means each final reproductive cell ends up with only 1 chromosome from each original pair. The chromosomes are literally physically sorting themselves into distinct reproductive packages.
Section 4. Comparing genes and chromosomes.
Now let's play a quick game of spot the difference between Mendel's factors and these new chromosomes. Look closely at column A and column B. Column A says it occurs in pairs, it segregates at gamete formation, so only one of each pair goes to a gamete, and independent pairs segregate independently. Now for column B, it says it occurs in pairs, it segregates at gamete formation, and one pair segregates independently of another. Notice anything? They are virtually identical descriptions. This was the massive aha moment for Sutton and Boveri. One column describes the theoretical behavior of Mendel's genes, the other describes the observable, physical behavior of chromosomes during meiosis. The parallel was just way too perfect to be a coincidence. Chromosomes had to be the physical vehicles carrying the genes.
Section 5. Morgan's fruit fly proof.
Enter evolutionary biologist Thomas Hunt Morgan. He wanted to take this from theory to undeniable, experimental fact. But to do this, he didn't use Mendel's pea plants. Instead, he chose a tiny, winged laboratory partner, Drosophila melanogaster, better known as the common fruit fly. Now, it might not look like much, but this little insect was about to completely revolutionize modern biology.
So, you might be wondering, why choose this incredibly unglamorous bug? Well, the fruit fly was the absolute perfect model organism for genetic research, for some very highly practical reasons. First, they could be grown in simple glass bottles on basic synthetic food, cheap and easy. Second, they complete their entire life cycle in just about 2 weeks. Plus, a single mating produces a huge, actually a massive number of offspring, which is absolutely perfect for statistical analysis. It's also incredibly easy to tell the males and females apart, and crucially, they have very clear hereditary variations, things like different eye colors or wing shapes, that Morgan could easily see with just a low-power microscope. By tracking these variations across thousands and thousands of fast-breeding flies, Morgan could literally track the chromosomes as they were passed down from generation to generation.
Morgan's experiments with fruit flies proved absolutely unequivocally that specific genes were linked to specific chromosomes. It completely validated what Walter Sutton had realized years earlier when he stated, "The pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried." Sutton brilliantly united the knowledge of chromosomal segregation with Mendelian principles, but it was Morgan's fruit flies that provided the undeniable physical evidence. This wasn't just a theory anymore. The physical basis of genetic variation produced by sexual reproduction was finally understood.
By discovering that chromosomes carry our genes, these scientists successfully bridged the gap between the invisible laws of heredity and the physical reality of our cells. Morgan's work even led to the discovery of genetic linkage and recombination, which opened up entirely new dimensions of biology. But this whole story leaves us with a pretty fascinating thought to take away from today's explainer. Think about it. If studying the chromosomes of a tiny, simple little fruit fly could unlock the fundamental physical basis of variation for all living things, what vast complex secrets are still hiding within the billions of base pairs of the human genome today? The mystery that Mendel started is far from over. In fact, finding that trapdoor was really just the beginning. Thank you so much for joining me, and keep exploring.